please afterwards ask questions for our visitor and also talk among yourselves and so on. It's so great to have Bill Crown here, who is heading up a really quite extraordinary new research effort, the Optum Labs, which I hope you'll tell us a little bit about it. It's a very interesting experiment in bringing rationality to medicine. And Bill was previously at the Thomson Reuters MedStat in their research and pharmaceutical division, and before that was a faculty member at Brandeis University at the Heller School. And before that, got his degree at MIT. So he's a long-term friend of this part of the universe.
Yes.
And we are so pleased that you're here and you're going to tell us about the identification problem. You didn't say and its solution, but we'll find out whether that second phrase is in.
A solution has been proposed, but thanks very much, Jeff. Thanks for having me in. I think I'll start out just by talking about a little bit about Optum Labs as an example of kind of the expanding access to data. That's just remarkable how much data is being aggregated and the potentials for beginning to link data from various sources together. My training is in economics, and when I was at Brandeis, I taught the stats course, statistics sequence in the PhD program there in the Heller School. And so I spent a lot of time thinking about estimating treatment effects, and treatment effects in the sense of some sort of a intervention or some sort of a program. And it could be a medical intervention, it could be a drug, it could be a way you're organizing care, like accountable care organizations or bundled payment models. All those things are treatment effects. And but we really have been in this observational data world kind of defined by defining the problem in terms of the data that we have available. So a lot of research has been done with these big medical claims databases, which are very rich in terms of the kinds of information they have. But one area they're particularly weak is in clinical detail. So if you want to study something like treatment effects in diabetes or cardiovascular disease or oncology, where having measures of both clinical outcomes and clinical severity as controls, you just don't have that in claims data. So as an economist, I think of that as a missing data problem, and that's going to generate biased estimates. That's called the endogeneity problem. Endogeneity is what economists refer to as anything that causes a correlation between a treatment effect and an error term in an equation you're trying to estimate. And it's very, very similar to the idea of confounding in epidemiology. They're essentially the same idea. So economists came up with this method of estimation method back in the 1920s, actually, in agricultural economics. It appeared in an appendix of an agricultural journal to try to control or to do something about the identification problem. The challenge they faced was that they were, trying to estimate markets, demand and supply in markets. But they only had market clearing prices and quantities. So you look at something like the wheat market, and you had market clearing prices and quantities, typically over time, but that's all you had. And they discovered that by bringing in additional data, data on things like rainfall and days of sunshine and so forth, which would influence supply, it actually traced out the demand curve. And similarly, by bringing in additional data on the demand side for things like, disposable income and so forth, it would trace out the supply curve. And so that kind of data augmentation strategy in combination with these statistical methods came to be known as instrumental variables, or the way economists have been kind of tackling with these sorts of data problems that have this statistical structure for almost 100 years now. So there's a lot of literature, and I, very early on, I was using a lot of this work in outcomes research, where it hadn't been used very extensively, and the methods have really been developed mostly in the labor economics literature. But labor economists became sort of concerned about the performance of these methods and the fact that you actually may do worse using these methods than ignoring the problem altogether. So I'm going to finish up with some simulation work that I did with David Van Ness at the University of Wisconsin and a colleague of mine in the life sciences business at Optum, where we knew what the truth was by design, and then we compared instrumental variables and regression models under various breakdowns and assumptions. So I'll show you those results. So Optum Labs is a research and collaboration and innovation center jointly founded with Mayo Clinic. And Mayo is kind of the first partner in the labs. AARP has announced that they are joining the labs several weeks ago. And we have about 10 or 12 other organizations that we're in discussions with. We hope to be able to announce maybe 6 of them before the end of the year. And the idea is to get people coming together with their different perspectives in tackling big problems in healthcare. And then also providing the data infrastructure and providing access to that data in a secure environment so that it can facilitate that process. So we set up Optum Labs because we felt that that kind of collaboration wasn't happening very much. You sort of get academia working with life sciences companies, as we can see all around us, and academia working with, and consulting firms working with the federal government and so forth. But you rarely get life sciences companies, academia, federal government, provider organizations actually coming together to, to craft a problem, to set out a problem and to look at it. And so, for example, if you're thinking about a payer like United Healthcare and how they would view the value associated with a particular pharmacotherapy or biologic, they're going to have a very different perspective on that than the life sciences company that's producing that is. But there is a common ground. So if you sort of get people to think about that and kind of tackle it on that common ground perspective, it has a big impact on how you craft the question in the first place. There are most of the work that's been done using observational data to look at things like variation in care has been done with the Medicare 5% sample, because academics have had access to that. But access to commercially insured data has been much harder to get. So life sciences companies have had it, but academics by and large have not. And government agencies haven't. So we want to be kind of an aggregator of a lot of data. I'll tell you a little bit more about what kind of data we have. And then electronic health record data in large scale is another issue where so much of the data is just, it's not research quality. You have a lot of data in small pockets in specific sites. Maybe 70 or 80% of the value of the data is in the clinical notes. The clinical notes aren't typically available for external research because of HIPAA-related issues. So creating data sets where you've got large volumes of high-quality electronic health record data that's research-ready, and then particularly if you can link that data to claims data, so you get kind of the best of both worlds, that's a big goal of ours. And then, so the first 2 top ones are research related, and the bottom ones are around prototyping and innovation, and just creating an environment where we can take these learnings that we get out. We figure if we end up publishing 300 papers in the next 5 years, that's not really going to be a success. be a nice success from an academic standpoint, but from the standpoint of actually having an impact on the healthcare system, that won't do it. So we've got to find a way to be able to pick projects that have a high potential for translation into clinical practice, and that's why Mayo is such a great partner, because they do a lot of this kind of translation work. And then ways to sort of take the findings and bake them into software tools and things like that will sit on the physician desktop, so that we're not waiting 17 years for something that appears in an article before it actually becomes standard of care. And then the last one is we're operating Optum Labs from a business model standpoint, really in a cost recovery mode. It's not like a consulting business. But getting cost recovery on data fees for research projects is never going to cover the cost of the lab. So it's a really big investment in terms of the infrastructure that's been made. So the real opportunities are around innovation and creating, the opportunity for access to data on the part of companies that need access to data in order to develop their service offerings or their products. And then the sort of an equity sharing sort of relationship. There's an opportunity downstream to actually do some significant cost recovery. But that would all be folded back in just in terms of covering the cost of the labs. So this will give you a sense of what we're working on in terms of Optum Labs. Virtually everything we're going to do is going to be a comparative effectiveness research question. And so it's the same framework that I described earlier in terms of the treatment effect. It's a head-to-head comparison of some alternatives. And they could be anything from drug treatments to different service delivery models and so forth. But one of the things that I think is really interesting, particularly for today, is that when you look at this data, whether it's claims data or electronic health record data, we've got the classic identification problem from an economist standpoint, because some of what we observe in the data is consumer behavior, and most of it probably is provider behavior. So you think about what's in there, things like procedure codes and diagnosis codes and all that. That's coming from the provider side, and then things like medication adherence, which you can observe in claims data in particular, is primarily a patient behavior. But these things are totally tangled up, and so that means that if you don't actively try to address that and pull those things apart so that you can understand how much of this variation is coming from providers and how much of it is coming from consumers. If you try to do something like develop, a copay model or reimbursement model for insurance for patients, it's undoubtedly going to be biased. And yet we've done that. We've got this huge national infrastructure that's based on sort of an implicit assumption that what we're observing in the claims data is consumer behavior. But it isn't. It's probably primarily provider behavior. And similarly, if we want to sort of understand levers that are going to influence provider behavior, we've got to pull out the piece of it, that variation that's due to the consumer. So this is kind of the classic identification issue. And that's why we're very interested in data linkage to enhance our ability to be able to tease these things apart, and then these instrumental variables methods. Variation in care, I mentioned earlier, that's a huge thing. Opportunity, there's 35 years worth of work that the Dartmouth Atlas folks have been doing that you could replicate with commercially insured data. So there's a lot of opportunity there. And then methodological research. My own personal interest is specific therapeutic questions are of interest to me, but sort of the really big thing that I'm interested in is creating the platform for better reliability of drawing inferences from observational data. There's so much noise about clinical trials versus observational data. There isn't any question that a randomized design is the strongest statistical design. But that doesn't necessarily mean that you can't rely upon estimates that come from observational data. So you got to understand the sources of bias that can come in from observational data, and then try to knock those out as much as possible. And to the extent that people have actually compared well-designed and conducted observational studies with quasi-experimental design, and compared those to results from clinical trials done on the same population for the same question, the results are remarkably similar. So we end up getting these differences when you've got all kinds of design issues around the kinds of populations that are enrolled in clinical trials versus those in observational studies. And then there's a lot of bad observational studies and there's a lot of bad clinical trials. So, you can't replicate clinical trials either, for that matter. So we're starting out with about 149 million lives of claims data going back about 20 years. A lot of data. And most of this is United Healthcare claims data. And then about a third of it is non-United data that's coming in from other health plans and organizations that Optum has relationships with. And we've also augmented the usual sort of claims data with other fields. So we've got, back in 2006, United was interested in developing high deductible plans. And they didn't want to create inadvertent issues in terms of access to care by race, ethnicity. So we went out and we got a whole bunch of data on social demographics, and we brought it in for diabetes patients. And we stratified the patterns of healthcare utilization for diabetes patients by these social demographic characteristics that aren't normally in claims data. And there were big differences in patterns. So we had done that on an initial draw of 2 million lives, and then I said, okay, go get everybody. So we've been bringing this data in since 2006, and whenever United acquires a new plan, that data is brought in, and then it's refreshed periodically. Most of it doesn't change very much, but some of it is. It comes from a marketing research company. So we do that as a business associate, not Optum Labs, but another part of Optum does that as a business. Yeah, exactly, company like that. So then 2 days after, Optum Labs was announced January 15th of this year. And 2 days later, we announced the acquisition, or actually we didn't announce it, we completed the acquisition of a company called Humedica. And Humedica is a company that goes into provider organizations and has an application that sits on top of the various electronic health record systems. So they'll go into like a Mayo Clinic. They tend to be about 70% of their data is from integrated delivery networks. And they hook people up with a unique ID across these different electronic medical record systems. And then they have really pretty sophisticated natural language processing that they do of the clinical notes to pull data out of clinical notes and put it into structured fields. Because often even things like blood pressure and height and weight, which are supposedly in the structured fields, the data isn't in there, and the physicians just write it in the notes. You've got to be able to pull it out. So this is the kind of stuff we've got. Recently, we got about 2 million lives of health risk assessment data, height, weight, and tobacco use, and so forth. And data just keeps getting richer and richer. These are the kinds of data.
They're good at telling you tobacco usage?
No, but some of them do. So there's no doubt a selection bias in terms of who reports that and who doesn't. And so these over on the your right hand side, those are the kinds of fields that are pulled out of the notes and put into structured fields. So there's a lot of work here. Cancer is one of the areas where Humedica has not really developed the NLP yet. They've done it in about, I think, 12 other disease states. So cardiovascular, mental health, congestive heart failure, diabetes, a bunch of them like that, but not oncology yet. Not yet. We're trying to figure out from a HIPAA de-identification standpoint how we can do that. oops. Try not to screw it. Thank you. So Mayo has a lot of genetic data, but we haven't imported it yet. So it may be that the way, so just as we don't take the notes from Humatica, but we take the data extracted from the notes and put into structured fields, I think that is probably the strategy eventually that we'll be able to use with genetic data, where you can take certain elements and then extract that. And so then when you bring that data in, we'll need to trade off against other fields in terms of which other fields are masked in terms of preserving the identification in the data. Our basic strategy is to do the linkage and then provide a research environment for people so that they can come in and have a virtual machine. They don't take the data off our server. But depending on what their research question is, we preserve the detail necessary for that research question and we mask the other detail. And that means they can do anything they want with that data within that environment. They don't need to worry about re-identification issues. And because they're not taking the data off, it reduces the risk of them matching against external data and so forth. So bringing in genetic data, you would just need to make the decisions around what other fields to mask. So I talked about this a little bit already. Claims data are really good for getting data across treatment settings and just because for all the stuff that's covered by insurance, you can see the claims for their visits to the doctor, all their prescriptions, ER, the hospitalizations, and all that sort of thing. But they're not very strong on the clinical side. There's a lot of coding biases in clinical data, in claims data rather, that are not as severe in electronic medical record data. We can see when we look in the electronic medical record data, there's better longitudinality in it because people tend to stay with their provider. So even if they switch health plans, they try to keep the same provider typically. There's a lot of much deeper data, unstructured data in the electronic health record. But with just an electronic health record alone, it's not very good for health economic questions. And the ability, you have a different sort of selection bias often in the electronic health record data because take something like mental health, for example, in a cardiovascular center, you have deep data, for the cardiovascular condition of the patient, but you don't know necessarily whether they have comorbid depression. And we know that when you look at people with and without depression who are otherwise similar, the outcomes are very, very different. And the healthcare utilization is different. So this ability to be able to get a broad view of the patient and then the deep clinical content that's where we want to go to the extent we can. So we talked a little bit about the hierarchy of evidence. Randomized trials are at the top. Quasi-experimental design with observational studies is probably the best. We can do it with observational data. The kind of pre-post with no controls is remarkably common in observational data and not very reliable at all because so many different other things are happening and you ascribe it all to the treatment effect. This is very common in evaluation of disease management programs, for example. Most of the vendors do not report data that's been with a control group. So they just do, here's what we saw for healthcare expenditures for these diabetes patients before we put in a disease management program, and then here's the cost afterwards. And then the return on investment is typically something like 6 to 1. But then when you do a control group with that, you're lucky if you break even. So it's having the control group is very important. So there's I think my own view is that there's a huge irony with the selection bias issue in terms of clinical trials, just because, who's in clinical trials? They're very selected patient populations. They're followed for a short period of time. Rare events typically are not captured very well because the sample sizes aren't that large, except for things like cardiovascular trials. So we do these trials for drug approval, and then the drugs get on the market, and they're used in totally different populations that have a very different clinical profile. You take those results that are so different in the real world, and you compare them to clinical trials, and ironically, people say those real world results can't be right, and it's apples and oranges in terms of the patient populations. Really, it's this difference about clinical efficacy versus effectiveness. So there's this difference in design versus the data quality. So my view is when you look at this and you think about the sources of bias in an observational study, most of the issues come from measurement error. So you're either missing variables or you're measuring variables with a lot of imprecision. And those two things, along with other things like wrong functional form and so forth in your equation that you're trying to estimate, all introduce a correlation between the treatment effect and the error term in the equation. That introduces bias. So if you can get better data quality, collect the variables that are needed to answer that particular question and have good quality data. So this is the issue with electronic health record data. You got to use the NLP to extract the variables and put them into structured fields so that they're available as control variables. Link that to claims data. A lot of it is data preparation and getting good data. And then you should be able to get really fairly similar results when using observational methods. So we've got very, good methods for controlling for observed variables in data. And the challenge is no matter how much we do, we're never going to have every single variable. We can never be certain that we've measured every single variable perfectly. And in fact, I can't imagine that's ever going to be true. There's always going to be sources of error that are coming in observational studies. So that's the endogeneity problem. What do we do about that? And so this is the area that economists have thought a lot about. And in contrast to epidemiologists who worry a lot about it too, from a confounding standpoint, and really try to deal with it from a research design perspective, economists have developed these methods, these statistical methods that try to deal with it. And I'll give you some description of that. So I've talked a little bit about this already. Basically, any of the problems associated with observational studies end up getting captured by the error term. And so from a technical standpoint, if we've got any variable xk where its correlation with the error term is not equal to 0, we have an endogeneity problem. So here are some examples of endogeneity in observational studies. Let's say, here we have a sample above the line that includes all the observations above the line. So we've got a group, maybe we'd like to draw some inferences about the whole population in the United States. Like, what's the treatment effectiveness of a particular intervention for diabetes, for example, in all the population in the United States, but we have only the commercially insured population. If we try to draw inferences about that, what will happen is that the regression line will pass through those observations that we have to minimize the sum of the squared errors, and we'll get this line, this gray line. But now suddenly, if we get CMS to come in the door and say, yes, you can have all of our Medicare data as well, and you get that population, and you estimate the line for the pooled population now, rather than this truncated sample, you'll get this line. And the difference in these lines is the bias. And this is the bias associated with having a measurement error in the sense of only having that population above the line. So that's likely a common problem. Years ago, I was involved in doing work in bipolar disorder, and I was working with a pharmaceutical company, and they were interested in how events leading up to, particularly drug use patterns leading up to um, hospitalization, were influencing what drug somebody was on. And what was really interesting is that the mean number of prescriptions for bipolar patients, this was in a commercially insured population, was 16. They were on 16 different medications across. And so if you think about what happens, and of course, the more medications they were on, the more likely they were to have had a hospitalization or emergency room visit with a mental health diagnosis. But what happens when somebody gets hospitalized? They get hospitalized, and the treatment protocol is to strip them off the medications. So you take all the medications away, and then you add back, and the person leaves the hospital maybe on 2 medications. So those things are going like this in different directions. One is it's very much like the supply-demand equation. So if you just look at the data and you estimate a model without trying to understand that, they could either cancel each other out, or one would dominate just because there's more people that don't go into hospital or something, and you just get a weaker estimate, and you get a biased estimate of the effect of those 2 different processes. So simultaneity is another source of error in observational studies. So one solution to this is, if we have a variable that's missing, is to go out and get it. So if we have claims data, and we really want to study diabetes, and we need hemoglobin A1Cs, and we want all the kind of cardiovascular risk factors, and we want their blood pressure, and height and weight, and all those kinds of things. That's all going to be in electronic health record data. We can go get it. That's probably the best thing to do, is to actually get the variable. Sometimes we'll have something where we don't actually have the variable, but we can kind of come up with a proxy for it. So we might hypothesize, for example, that somebody's the wages that they get are associated with their intelligence. Well, intelligence is pretty hard to measure, but we could say, well, IQ is our proxy for intelligence, and we're creating kind of human capital around intelligence by their education, and those things are a way of kind of controlling for their ability. But economists have a method that they call instrumental variables, which essentially is a way of using all the variables that you have to create a measure for the variable that you don't have, and then using that in a statistical model. So Slock and Treby did a review of trying to find the origins of instrumental variables, and their earliest reference they could find to it was an agricultural economics article back in the 1920s. The idea is to find a variable that we'll call it Z, that has a correlation with the endogenous variable. So let's say we have we're trying to estimate a treatment effect. We want to find something that's correlated with that treatment selection, but isn't correlated with outcomes. So in some of the work that I did in depression, I ended up coming up with something which was for the drugs that were being compared, what was the relative time on the market that those drugs had been on the market in days, and I took the ratio of those days at the time when the drug was being prescribed, and that was a measure of a measure of diffusion of knowledge about side effects and clinical effectiveness and so forth in the real world. and it was predictive of what drug somebody got, but it wasn't correlated with outcomes. It wasn't correlated with health expenditures for depressed patients. Well, those are really hard to come up with. It's very hard to come up with variables that are correlated with the problematic variable, the endogenous variable, but uncorrelated with outcomes. That's the challenge. So the way economists did it initially was to take that variable, say it's a treatment variable, and regress it on everything else. So they just took all the other variables that were exogenous and they ran a regression between that as the dependent variable and all those others. And they didn't really care about the coefficients or anything like that. But what they wanted to do was predict the value for that endogenous variable as a function only of those exogenous things. Because what it does is if you create this new variable that by definition is a function only of things that aren't endogenous, it's a linear combination of things that you have assumed are not endogenous, then it has to not be endogenous anymore. And then you can use that instead of the original variable as a variable. So an example would be we would take drug choice for antidepressants, for example, and we would run a regression with drug choice as the dependent variable and all of our explanatory variables predicting it. But then in our outcome equation, instead of using the dummy variable for the drug, we would use the predicted probability that that patient got that drug. And by definition, that severed that correlation with the error term. So it's a very interesting idea. It's a statistical kind of fix for this endogeneity problem. And Hausman back in 1983 came up with a couple of different tests, one in 78 and one in 83. But the idea basically is to know whether or not you've got an endogeneity problem. The challenge is whether the residuals are correlated with the outcome. So if I have my drug equation that I'm trying to predict in order to get my instrument, that drug equation is going to have an error term on it. So when I go to estimate my outcome equation, if I take the errors from that drug equation and I put them in the outcome equation, errors should be random. But if they're not random, if there's a significant coefficient on those errors, what that means is something is amiss. There's variables missing, or you've got a wrong functional form or whatever, so you've got an endogeneity problem. So it's a very, very clever idea that you just use the errors themselves as a measure for what's missing. And if there's a correlation between the errors and the dependent variable of interest, you've got an endogeneity problem. But not only that, but the fact that you've got the errors in the model takes the endogeneity out of the treatment variable. So it kind of peels it back out. So it not only gives you a test for whether you've got an endogeneity problem or not, but it also corrects for it at the same time. Wonderful idea. This is why, it's so enthusiastically embraced. So I already talked about the supply-demand equation. So the idea of this identification is we need enough information to be able to tease out these separate relationships that are all bundled up in the data. And you don't hear people talking a lot about this. There's been some nice work done by some economists in the medical school over at Harvard. Hayden Huskamp had done some nice work where she was looking at relative prices and thinking about stuff this way. But in general, people just run regressions on the claims data or the electronic health record data and aren't terribly concerned about the identification problem. By the way, I got into this work because I was concerned about the way propensity score methods were being used in a lot of the early literature, where people were using the predicted probability of somebody getting a drug or not and putting that probability in the outcome equation and propensity score. And I said, that's just an inferior version of instrumental variables because they're not worried about whether it's identified or not. They're just plunking it in there. So I gave this paper at International Health Economics Association meetings, kind of aggressively asking the question, is propensity score just an inferior version of instrumental variables? And it just got me thinking about instrumental variables in a much more critical way. So I've actually come around to the fact that I think propensity score is a better way to go, but we'll get to that in a minute. So there's lots of examples of now of the use of instrumental variables in the health economics literature. And the most famous citation is McClellan, McNeil, and Newhouse, who used distance from cardiovascular centers as an instrument for sort of access to care and severity, because people that are really severely ill and are having an AMI are likely to go to the local hospital, but that's not a very good measure of whether or not the intervention is a more intensive intervention is successful or not, because it's confounded by the fact that they're just being taken to the nearest place. So the idea was to try to control for that and tease it out so that you could measure the whether going to a cabbage facility, for example, actually resulted in better outcomes. So not surprisingly, distance is very often used as an instrument in outcomes research. I mentioned that this instrumental variables came out of this kind of labor agricultural economics literature originally, but those data were very simple. They were interval data, regular regression models, and so forth. And so what's happened as claims data began to become available in the late 70s, early 80s, we're kind of wrestling with these discrete choice problems of where you've got 0 1s for a dependent variable, somebody gets a drug or not. You've got lots and lots and lots of data, a lot of variables. And those are not kind of the simple ordinary least squares problems. But they were very similar. These outcomes research questions were very similar to the problems that economists were wrestling with in the labor economics literature in the mid-70s. So, there the challenge was the economists were kind of naturally interested in whether or not, or what the relationship was between the number of hours that people were willing to work and the wages that were being paid. But it was like that censored sample example I gave you. You were only observing that for the people that were in the labor force. You're only observing the wage hours relationship for the people in the labor force. And so economists said, well, what if it's the fact that some people aren't working because the wages aren't being offered, that aren't even high enough to entice them to be in the labor force? And this became known as the reservation wage. What is the wage that would be needed in order to entice people to go into the labor force? But in these big national probabilistic surveys from Bureau of Labor Statistics, what you had samples, and you could observe whether somebody was in the labor force or not, and you had data on the whole population, random samples of the whole population, so you could model whether somebody was in the labor force or not, just like that drug selection equation. And you had things like, marital status and income and numbers of kids at home under the age of 6 and things like that, and you could model that, and then you could subset down on the patients that were in the, employees that were in the labor force, and for those employees that are in the labor force, you could get those error terms from that selection equation and plunk those in. So that was the idea. And this, so it started to become applied to labor economics. So when I left Brandeis and I started working at MedStat, I was doing policy work on Medicaid expansions for the aged and disabled. And I got a call out of the blue one day from somebody in the MedStat pharmaceutical consulting business. They said, do economists have any methods for dealing with sample selection bias? And you could sort of see this the same statistical problem for the outcomes research as it was for the labor economics. So I spent the next 15 years doing application of econometric methods to outcomes research. But there was a lot of times when I got questions around from reviewers around endogeneity. So the reviewers would come back and I'd have run a regular regression model, and the reviewer would come back and say, well, there's these potential sources of endogeneity. Did you test for it? Did you correct for it? And you go off and you do instrumental variables and you get almost the same answer, but the standard errors were bigger. And what always got published was the more sophisticated method, the instrumental variables method. So the question was that really the right answer? And particularly because there's been a lot of evidence from labor economics that these instrumental variables methods have a really, really big standard error. And there's some other issues associated with them. So how well do they really perform in actual practice? So my very first example in the antidepressant work was comparing SSRIs against tricyclic antidepressants. And Group Health of Puget Sound was doing a clinical trial on this question, and we were doing some work for Eli Lilly at the time. And they said, well, do you think the FDA would ever take this as, if we did one database study and we did this clinical trial in Group Health of Puget Sound, we got similar results, do you think the FDA would sort of take that as two well-controlled studies? I don't know. I didn't know anything about this business. But I said, I think it's appropriate to sort of do these kinds of methods. And so we did, and we modeled that. First equation is modeling the drug choice. So whether or not somebody got an antidepressant, tricyclic versus an SSRI. The second one was the drug use pattern. So how many days did they stay on the drug with drug as an explanatory variable now? And then I ran separate models for the SSRI group and the tricyclic group, which would allow drug basically to interact with every single variable. And what we found when we looked at the data was that this was kind of the classic case where at the time people were talking about the evidence from the clinical trials. And the evidence from the clinical trials was that the treatment effect for SSRIs was essentially exactly the same as the tricyclics. There was about a 50% response rate in the clinical trials. And so the big prescription benefit manager companies were saying, well, why would we ever reimburse for an SSRI that costs 10 times tricyclic when they've got the same clinical efficacy? But when you looked in the data, the patients that initiated on the SSRIs were much less expensive in terms of total healthcare costs than the patients that initiated on the tricyclics. Interestingly, they were almost exactly the same in terms of any claims that had a mental health diagnosis on them. So, virtually a dead heat. But where you saw the difference was in the non-mental health. And so actually these graphs were very helpful to us because as we thought about this, how does somebody get into treatment for depression? Well, they go into the doctor and they're complaining about headache, complaining about the fact they're not sleeping well, they've lost their appetite, they've got back pain. Finally, the doctor says, I bet they've got depression. And they put them on an antidepressant. So to the extent that somebody stays on the antidepressant longer to resolve the underlying depressive symptoms, where you see it is actually in the non-mental health claims, because all that stuff is showing up in the database as medical expense, but not mental health expense. So I learned two things there. One's the difference between, well, a number of things. Difference between effectiveness and efficacy. The fact that you can't look just at the disease-specific costs. You've got to look at the broader costs. And think about it now in terms of the, trying to analyze healthcare outcomes for medically complex populations, which is a big research area, or trying to understand healthcare utilization and outcomes for the elderly, which have all these comorbidities. You've got to have this broader view of the patients. So the upshot was that we did find selection, but we found it with the tricyclic group and not with the SSRI group. And I think the reason was that there's an issue with respect to tricyclics in terms of suicidality potential. So people could take them and to commit suicide. They're dosing specific. So the psychiatrist would be familiar with the dosing for tricyclic patients, whereas primary care docs would be much more comfortable prescribing the SSRIs. And so I think as a result, the sicker patients tended to be getting the tricyclic antidepressants because they were seeing psychiatrists. So that was the hypothesis. And that's why we set it up where with the drug choice, one of the explanatory variables was the provider type, whether it was a psychiatrist or a primary care doc, and then the drug use pattern, because we felt like the drug use pattern was really going to drive healthcare outcomes, and then separate equations for the two groups. Yep. I think I'm being timed out.
Just restart. Seems fine. Okay.
So this last little bit is just going to give you some simulation results where I basically kind of invalidated 15 years of my own work. But Brandeis's motto was truth unto its innermost parts. So I tried to do that. So let's see if I can skip through these. I should stop and give you a quiz. Okay, so one of the, so there was a literature that showed that weak instruments can not only lead to bias, but also not only large standard errors, but also bias. There's quite a large literature in this area, mainly in labor economics. But there was a theoretical paper that was written by Bound, Yeager, and Baker in 1995, where they basically proposed a relationship between the strength of the endogeneity problem, the strength of the instruments, and whether or not there was still residual correlation of the instruments with the error term. And they provided this kind of graphical analysis of this. And so basically what we did was we just replicated the Bound, Yeager, Steger, Yeager, Bound, Yeager, and Baker results with an empirical simulation where we knew what the treatment effect was, and we just went right through their paper. We also looked at sample size, which is important in the big data environment. As it's not, I would just say that it's not generally understood in the big data world that more data doesn't solve the bias problem. So having more and more and more and more claims data is not going to solve the bias problem unless it helps you to link to data that are missing. Because otherwise, actually, you just end up getting really precise measures of bias in that case. So we wanted to estimate the treatment effect, and we've got some conversion issues here. But the this first equation on the upper left-hand side is the correlation between the treatment variable and the instrument. So we're saying we're going to start with an example where we've got a 40% correlation between the instrument and the treatment session. Really, really strong instrument. So this is a kind of a best case scenario in terms of instrument strength. And then we're also going to take an example where we've got a really, really bad endogeneity problem. So we've got a correlation of 40% between the treatment variable and the residuals in the outcome equation. So really strong instrument, really bad endogeneity problem. Exactly the kind of case when you should be using an instrument. But we're going to start out with a smaller sample. So in this first example, we're assuming there's no correlation between the instrument and the error term. So we've basically got a perfectly clean instrument. So in the case of a perfectly clean instrument, if there's no bias, the difference between the true value and the estimated value is going to be 0. So for any particular sample, we could be here, we could be here, we could be here, we could be here. But the sampling distribution, we want it to be centered over 0 in order for our estimator or our statistical approach to be unbiased. And sure enough, instrumental variables, which is the blue line, is centered right over 0. So in the case where we have a perfectly clean instrument, we have no bias and it's centered right over 0. No bias and it's normally distributed. In the case of ordinary least squares, where we have completely ignored the endogeneity problem and just run the treatment effect estimate, which in a sense is what we're doing when we use claims data alone, and we're not trying to correct for missing variables. We have a biased estimate, but notice that it has a smaller standard error, but it's still biased. So in this case, we would probably, I think we'd pick everything else equal, not knowing which sample we had, where we were on these distributions. We'd want an estimator or an approach that was going to give us an unbiased estimate on average. So instrumental variables looks pretty good for this case. Now, in this upper panel, if we have an 8% residual correlation between the instrument and the error term. So it's not a big correlation, but an 8% correlation between the instrument and the error term. They're both equally biased. So both ordinary least squares and instrumental variables are now biased. Instrumental variables has a bigger standard error associated with it than ordinary least squares was. But again, it's not really obvious which is the better method, because all these estimates in the area under the curve here is probably bigger in instrumental variables than it is with ordinary squares. But it's kind of a coin flip. Now, if the correlation isn't 8%, but it's 16%, actually, instrumental variables is even more biased than ordinary least squares was. And this is completely ignoring this really strong endogeneity problem. So we've got a 40% endogeneity problem, completely ignoring it, and we're doing better with ordinary least squares than we are with instrumental variables because of this variability problem here. And so this is of concern because it's so hard to find these instruments that invariably economists are leaning over backwards to find these instruments, and they're always weak. And because they're weak, it raises the chances that you've got residual correlation because you're not really controlling for everything. So here's exactly the same situation, but we just upped the sample size from 200 to 2,000. And so we have unbiased instrumental variables, biased ordinary least squares, but really tight variance, They both have tight variances. And so this is the point about bigger samples don't do away with the bias. What it's done is it's actually completely separated the distributions. So in this case, what it means is that you just have a more precise, measure of what the bias is, which, that's not a bad thing, but it doesn't solve the bias problem. So here's the case where we just change the, we cut the degree of the endogeneity in half, and we also cut the degree of the strength of the instrument in half. So we went from 0.4 to 0.2 in each case, and we go back to our 200 sample. But look what happens when the instrument isn't as strong anymore. When the instrument is only half the strength, it just flattens right out. So now, with any particular sample, your chances of being off in the tails with instrumental variables with a weak instrument are really high. And you could do a lot worse, with instrumental variables than with ordinary least squares. We see the same general patterns, but notice that we cut the endogeneity and strength of the instrument in half, and the thresholds that create these challenges here are also cut exactly in half. This is what Baumjager and this is exactly what they had proposed. This relation, the strength of the instrument, the strength of the endogeneity, and the strength of the residual correlation would have this relationship. But I would argue this is probably a more realistic case in terms of the instrument strength, because we generally don't have really strong instruments. And then here's what happens with 2,000 observations, and then with 10,000. You just it just gets more and more precise. So the bottom line is that I think we're sort of in a new world, in terms of our ability to do good observational studies and link data together, to be able to tackle, these challenges. Any observational study is going to have these challenges that we've been talking about, but our ability to attack them is so much greater than it has been. And it's just, these data cooperatives are just springing up all over the place. I think even though I've definitely been negative with respect to IV, what I'm doing as a result of that simulation work, I'm really kind of focused on data, because I'm not very sanguine about the ability for statistical methods by themselves to overcome, severe data limitations. I think we've got to focus on the data. But then the methods themselves are still valuable because they give you a test. If nothing else, they give you a test for whether you've got an endogeneity problem or not. And that's a really important thing to know, because, if you have that, significant coefficient on that error term variable that you've got in your outcome equation, it tells you, I should be cautious about the strength of the evidence that I'm, getting from this study. I think one final area that is very interesting is, what do you do when you can't link the data, or you can't link all of the data? So I mentioned that in Optum Labs, we've got this 149 million lives of claims data, and then it's actually about 37 million lives of electronic health record data. And we know that they have an overlap of about 15%, because we've looked. We're now linking that overlap, that band together. So we're going to have 5 or 6 million patients where we've got all the claims data plus all the clinical information that we have from the Humedica database. But you don't want to throw away 145 million lives of claims data and 32 million lives of electronic health record data. So what can you do? I mean, does it have to be the same people? Can you impute clinical distributions, for example, multiple times? to the people in the claims data, and then be able to have those distributions of clinical distributions as control variables. And if you do, what are the properties of the estimates that you get on the super sample versus the subsamples? And so what methods are the best methods and so forth. So one of the areas that we're very interested in is kind of the statistical infrastructure around, around data linkage. Because it's interesting, when you look in the literature, most of the literature is focusing on the errors that get introduced by imperfect linkage. But that's kind of a secondary question. The real question is, the reason you're doing the linkage in the first place is to reduce the bias by just having just a select set of the data alone. So we really want to look at what's the bias, you know, if you have claims data alone, what's the bias when you have the clinical, and then how does that get affected by imperfect linkage and by, you know, these multiple imputation techniques and so forth. So with that, I think I'll stop and see if you guys have questions.
So do statins make people live longer?
You know, I think certainly from the standpoint of the evidence was flawed in terms of the modeling that was done for that question. Because the models, you know, just couldn't replicate the mortality or event rates, and they overstated the event rates by about twice, which interestingly was the amount by which they recommended the expansion of the statins into the population was about twofold. So I'm not sure that, I think there's a lot of evidence that statins certainly save lives for the population that should be on them from the Framingham Heart Study, which is, of course, an observational study. But I'm not so certain that there's a lot of convincing evidence that they should be dramatically expanded.
The actual data issues with observational data and statins, you're interested in applying these methods?
Yeah, you know, cardiovascular is one of the areas where I think there's a lot of potential. So, you know, we've had the Framingham Heart Study data, but these big observational databases have a lot of data that probably is not in the Framingham Heart Study around healthcare utilization and broad-based medical comorbidity. And, you know, I think one of the areas that's really challenging from the standpoint of medicine is what do we do with these patients that are medically complex? So we've got guidelines that tend to be, they're disease-specific guidelines, cardiovascular guidelines, diabetes guidelines, guidelines for treating asthma, and so forth. And so they're disease-specific guidelines, kind of treating the patient in the silo. But if you take cardiovascular patients, for example, this is kind of the classic, group that's likely to have metabolic syndrome. So they're likely to have cardiovascular issues, blood pressure problems, obesity, hypertension, and hyperlipidemia, all these things together. And then to boot, they may have depression, or they may have, breast cancer or whatever. So what do you do with a patient like that? We don't have any guidelines for that. So a physician treating a patient is going to treat them right now. They're going to treat them for whatever it is they're seeing them for. And if they're a primary care physician and they're aware of all these other things, they're going to have a hard time figuring out what to do. Because there may be, if there's 3 conditions and there's 5 top priority things that the physician should be doing for each of the 3, there's 15 things. Well, which 15 should they do, and how should they prioritize those? So one of the things that's really interesting, I think, is I didn't talk about machine learning methods, but one of the things that machine learning does really, really well is put people into groups. So you could find patients that are really similar to the patient that is in front of the physician, and say, what were the treatment options that were, and do this in real time. Have technology that will do this in real time and present the physician with options and say, here are the things that have been done for patients just like this patient that's in front of you, and provide guide. So maybe that means that we go completely away from clinical guidelines at all, because as soon as you start getting into this medical, complex area, you can't have guidelines for all these things. Doesn't really make a lot of sense. What you need is a patient, a way to treat a patient with these conditions rather than treating these conditions. But what I worry about with the machine learning is that the machine learning will put patients into groups, and we've got sophisticated methods for analyzing treatment effects for those groups, but the machine learning methods don't worry about what's not in the data. So the idea, the way an epidemiologist or a health economist would think about estimating a treatment effect is they would be very concerned. Do I have all the variables? And if I don't have all the variables, how can I, deal with that? And whereas machine learning methods are just kind of, put everything together, put people into buckets, they're going to do that really well. So I think actually it's where the power may be is machine learning as a first step. Machine learning as a way to define the groups, and then the analysis of treatment effects within those groups using more traditional methods. Yeah, right. No, that's definitely true. Yeah. 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 And I mean, that's one of the things that, the machine learning methods do, by construction in order to avoid the kind of the overfitting problems is they create these, these groups and then create sort of test samples, learning samples, and then test samples, which is something that should be done, much more systematically and much more commonly in epidemiology and health outcomes, but it's not done that much. Yes. Yeah. Yeah, the bootstrapping methods won't solve the bias problem, but in terms of the work that we're going to do in the labs, where we're going to compare different methods, what the bootstrapping methods give you is a way of creating these empirical sampling distributions, like we did in the IV paper. So what you can do potentially is you can estimate the bias, associated with using claims data alone, how that gets reduced by bringing in the linked clinical variables. What sort of effect does it have when you bring in kind of the, you do multiple imputation for all of the missing cells that you have? And the bootstrapping is nice in that context because you can take each one of those and you can estimate them, 500 to 1,000 times, get a distribution, and then compare the distributions. And that's just not, it's not a huge deal to do that anymore with the kind of computer power we have. The machine that we have, for example, we have a Teradata box for this, and it will, we've got this query tool that we call natural history disease that allows you to be able to, with drop-down menus, specify inclusion and exclusion criteria for procedures and drugs that people have been on and so forth. And then right now it does a very simple match, exact match of people that didn't get that treatment or that don't have that disease, but it matches them by gender and by age and by hospital referral region and so forth. And so you look at the data and it looks like a propensity score. So it's all balanced. But we are going to extend that to include propensity score matching. But when you do this bootstrapping, or when you do this natural history disease on this Teradata box, first it's already kind of gathered all the codes in these drop-down menus, so you don't have to go running around the ICD-9 manuals and procedure code manuals and stuff to extract the codes and then put them into SAS or Stata or R or whatever it is you're using. Then do the ETL, this massive database, and wait for it to come back, and then write, programs to create the tables and so forth. That is like a 3-week process to do all that, and this natural history disease tool does it in about 12 seconds, hitting up against this, huge database. So
Is that the SAP machine or what's the gadget? What's the tool you're talking about?
the natural history disease,
Yeah.
it's a homegrown tool. Yeah. So what we're doing is we're offering for the partners in the lab, it sits within these virtual sandboxes so that they can kind of do rapid assessment of feasibility of questions. It's not the research tool to actually answer the question, but it helps you to kind of weed out a lot of stuff very quickly.
Thank you. Oh, yes, go ahead.
In terms of the data linkage issue and reducing missing variables problem, just wondering, have you been able to identify certain data sets that are more helpful in solving that problem than others, and were there any surprises or
Yeah,
I just think like the marketing
yeah, that's true. So one of the things that's interesting about AARP is they've got all kinds of consumer data. And with this marketing company, who shall remain nameless for the time being, we just got just a very limited number of variables, but they've got all kinds of stuff. So the challenge is if we bring in a lot of detail on what magazines people buy and, their expenditures on automobiles and cars and heat and all that sort of stuff. I think that's really important because I think people make decisions around health insurance and seeking health care and so forth in the context of a family budget. So bringing that data in, I think, is very important. And I think even what we have in terms of just the income variables and race ethnicity is very important. So I think that's a big one. And I think if I had to say 3, I would say consumer data, clinical data, clinical severity data from the electronic health records, and then the third is patient-reported outcomes. And the patient-reported outcomes, that doesn't sit in most of these data systems unless you're getting the data from a mental health clinic and they're collecting it PHQ-9 as part of their assessment of the patients and part of the clinical practice. But it's interesting because when you look at the healthcare utilization data, really high cost patients, 50% of them either had no cost or very low cost in the previous 6 months. So something like just a SF36 or an SF12 measure of health status gives you about a 15% lift in terms of variance reduction in predicting healthcare utilization because you're basically capturing some of the variance that you were missing before because if you didn't have the explanatory variables in the prior period using just the claims data alone. So I'd say those are the major domains. And there's a lot. So PCORI is obviously an area where there's a lot of focus on patient-centered outcomes and collecting data. But that data is going to have to be linked to this other administrative data that we have.